Hello everyone, welcome to the 14th APEC CMRA. My name is Sujin Lim and I am a biology teacher at Changdeok Girls High School in Seoul, Korea. Today, I would like to share some knowledge and do's and don'ts in science project or research. These are the contents that we'll go over today. I'm sure that students who are participating in this program have curiosity and interest in science. Scientific knowledge is gained through rigorous research, and you might have already done some research in science and will do many more in the future. Science is a way of approach to understand the natural world, and it developed out of our curiosity. Curiosity allows us to observe the natural phenomena and ask questions. So what questions do you have in this picture? When you have questions, you might want to have the answer for the question. Inquiry is at the heart of science, a search for information and explanation of natural phenomena by collecting data. When you look up the dictionary, you will find the definition of research. Based on the definition, research is more systematic process than inquiry to new information or understanding. Through research, you can produce facts or information rather than consume them. For example, people have wondered how plants can grow just with water and sunlight for hundreds of years. Scientists such as Kelvin, Benson, and Bazham tried to find out the pathway through which carbon dioxide in the air is turned into sugar in photosynthesis. Those scientists designed this experiment and collected data like this. Based on this data, they formed the Kelvin cycle and won Nobel Prize in 1961 in chemistry. Research is a careful, detailed, and systematic study of a phenomenon to learn something new about it or studying it from a new perspective. Scientific method is a way of conducting a research. Now let's talk about scientific method, a way the scientific research is done. Most of you are familiar with it, but I would like to stick to the basics. In research, scientific knowledge is gained through a nonlinear, dynamic process called the scientific method. The scientific method is not a strict set of rules to be followed, but rather a way of approaching problems in a rigorous but open-minded way. For example, while analyzing the experiment results, additional data collection could be needed. Also, at the analysis or conclusion steps, you can ask if the question was right or if the answer is right for the question. Or you can ask a new question after the conclusion. Let's look at the scientific method one by one. When you ask questions, Try something new rather than the obvious, and develop your ideas with your teammates and ask advice from your mentor. Before you form a hypothesis, you need to search for previous studies. As a high school student, it is very important to find reliable research articles and learn from them. But you still have to focus on your original study based on existing knowledge and previous findings. If you are preparing for a science fair, it might be helpful to look up some previous prize-winning results. Be sure to remember not to plan the experiment based on blogs, YouTube, or Wikipedia. Those sources are not always reliable. Usually, we tend to find interesting research results from secondary sources such as news, magazines, articles, reviews, or books. Do not start your research based only on secondary sources. Science magazines such as Scientific American, Popular Science, Discover, and National Geographic are advisable for secondary source for high school students. It is easily accessible online these days. For health or clinical matters, CDC, NIH, and WHO are good secondary sources. If you visit the Nobel Prize Organization website, you can get the general idea of the winning research and also advanced information for further studies. When you're not sure about the reliability of the science article, try the most cited journals in each science field. These are some of the most trusted primary sources, but they might not be as easy as you expect to understand. Then ask for help from your teachers or mentors. I would like to take an example of a primary and secondary source. When you search for the health benefits of garlic, you might find this website, which is a secondary source. When you click on the number of the research topic, it directs you to the research article, which is the primary source. This article is from Scientific American, and it took my attention because delaying the time of morning classes for high school students was a big issue in Korea back then. When you look up the references, you will find those primary sources. When you do web search, Google Scholar, PubMed, Archive are good sources. Some of the research articles are not free, but it might be free when you use your library account. If it's not, ask around your sisters or brothers at college. And don't forget to make a list of the references. When you look up some articles on humans, look closely how the research was conducted, because it's impossible to take into consideration every single variable when it comes to living organisms. There are ethical dimensions, but it is also a matter of accuracy. We cannot generalize the animal test results to humans due to uncontrollable variables, and differences among species. Forming a hypothesis is very important in research because experiments are designed based on a hypothesis. A hypothesis should be testable and falsifiable. It is quite extreme, but let's take an example of a hypothesis which is not falsifiable. The hypothesis is if someone is nice, he or she will go to the good place after death. In this statement, being nice is subjective and nobody knows what happens after death, so the dependent variable cannot be measured. This is another example. If cancer cells are not accessible to you, you cannot measure the division rate. A hypothesis is formed within the limit of your experiment equipment or devices. For example, if you are trying to conducting an experiment on enzyme activity, getting pure enzyme is hard or extremely expensive. Try to find the alternatives. People usually use potato, banana, or animal liver for catalyzed lab. Hypothesis usually is written like this. Y is the independent variable and Z is the dependent variable. Based on the hypothesis you formed, you can design experiments to collect data. Sometimes the word theory is misused in a context where hypothesis should be used. The picture on the right side explains French chemist and microbiologist Louis Pasteur's hypothesis. Some examples of theory in science are Darwin's theory of evolution or Einstein's theory of relativity. They are well supported by a much greater body of evidence. When the hypothesis is ready, you can design an experiment to collect data to test the hypothesis. Controls are used to demonstrate that the response in the test group is due to a specific variable. For example, you observed that there are more light-colored mice at a white sand dune beach and more dark brown-colored mice at an inland area. If you form the hypothesis in which there is a correlation between mice coat color and their habitat, how would you design an experiment? In a previous case study, to test the hypothesis, the researchers chose mice fur color as the independent variable and then measured predation event. When you look at the figure, the variables and the results of the study are easy to find. When you present the result in the form of graph, usually the independent variable is shown on the x-axis and the dependent variable is shown on the y-axis. In an additional experiment, the other independent variable was added and a single figure was used to show two or more data sets. This type of presentation is useful when comparing the trends for two or more treatments. These groups use the same measurement units and a similar range of values for the dependent variable. The two graphs must be distinguished with a key. Let me show you one example of a controlled experiment. Back in the 19th century, it was not clear that diseases could be caused by microbes. French chemist and microbiologist Louis Pasteur was the first scientist who applied vaccination in a controlled experiment to prove that the anthrax is caused by microbes. He developed vaccines against anthrax and rabies. Recorded observations are called data, and these are two types of them. Qualitative data relies on descriptions, while quantitative data relies on numbers. Many studies of animal behavior involve the collection and systematic recording of qualitative data. For example, Jane Goodall spent decades recording her observation of chimpanzee behavior. Quantitative data are generally expressed as numerical measurements and often organized into tables and graphs. Tables provide an accurate record of numerical values and allow you to organize your data so that relationships and trends are apparent. Scientists analyze their data using statistics to test whether their results are significant or not. Graphs provide a visual image of trends in the data in a minimum of space. This will help you to evaluate your results as you proceed and make adjustments as necessary. Figures are usually labeled below the visuals, while tables are labeled on the top. In science, quantitative data are preferable as it is easier to process, but not every data in science is quantitative. How can you turn the color of banana or strawberry to a degree or to numbers? For example, if you look up the color system, you will find that there are numbers that correlate to every color. So if you do a survey with banana colors, you can assign a number to each banana. When you do the survey, the sample size must be large enough to provide enough unbiased, reliable data. Even our moods can be analyzed with numbers. For example, you can show how happy you are from a scale of 1 to 10. Collecting data should be accurate and precise. Let's take an example. One student tries to find out the effect of antioxidant extracted from the inner peel of a tangerine. The antioxidant is water insoluble, but the student extracts it with water and checks the effect with it. Or somebody cultivates anaerobic microbes with the air. Can you believe the experiment results or not? To collect reliable data, use the right method with appropriate devices or equipments. Familiarize yourself with them. In biology, be careful about sample contamination. Sample size should be large enough not only to detect the desired effect reliably, but to observe the possibility of outliers. Also, don't jump into conclusions right after just one experiment. During the whole process, don't rely on your memories too much, but always take notes and record data. You should take pictures while collecting data, but it doesn't necessarily mean that you should show all the pictures you take. Select essential ones for presenting. Also, don't just take pictures from the internet for the result of the experiment. Pictures with high resolution are preferable for a better interpretation and understanding. Labeling samples will help you not to be confused or not to manipulate the data. If you can't find the equipment such as electron microscope and microbes for your research at your school, try university labs or hospitals. Don't wait until the last minute to realize that you don't have the right equipment. And after the experiment is over, don't forget to clean up by yourselves and return everything you borrowed. Experiments with animals in high school are banned in Korea now, but when it was allowed, students used mice or hamsters a lot. And once students left their mice cage in their locker in the science lab and went home on Friday. When they realized and opened the locker the next week, the cage was all empty. Now let's talk about sharing the results. You can share your experiment results as research papers, posters, and presentations. And we will talk more about that during live streaming class next time. Last but not least, let's talk about research ethics briefly. In general, do not falsify data and cite references properly and paraphrase previous studies instead of repeating them. Plagiarism is not acceptable. Model animal issues are important. Koreans have higher ratio of Helicobacter pylori in their stomachs. In 2001, Dr. Barry Marshall was the commercial face of a Korean brand of probiotic yogurt drink which is known to be helpful in treating in Helicobacter pylori. He was awarded a Nobel Prize in 2005 for his work linking stomach ulcer with Helicobacter pylori. Prior to his research, people just thought that stress and spicy food caused ulcer. To provide that the disease is caused by the bacteria, he even downed the liquid shot of bacteria to give himself an ulcer. He arranged an endoscopic biopsy on himself to confirm the diagnosis, treated himself with antibiotics, and was cured with no lasting effects. He was convinced. So is this a never give up attitude or violation of research ethics? Now let's talk about the work ethics. Most scientists work in teams. Good communication is important in order to work with your research partner and to share results through seminars, publications, and websites. What do you think is the title of this picture? Please work together with your teammates and don't hesitate to get some help from them. As a high school student, through research experience, you will face unexpected problems with every aspect of your research. Learn to be flexible and still persistent. These are the references. And thank you for listening. Good luck with your study.